Recording in progress.
You're mute, Boris. You're on mute. Is he intentionally on mute? Well, maybe it was intentionally. No worries. Unmute our room.
Yeah, Boris, sorry, I can ask you to unmute, but I can't unmute you.
Did you get it, Ed? Yeah. Can you hear us? Can you hear us now? Good. Yay.
We can't hear her, but we can hear you.
We've conquered technology again for now. All right. This is our second meeting. Thanks everyone for joining us. This is we are meeting for the Stanford Psychedelic Science interest group. So for those of you that didn't join us last time, we are doing this every about once a month, every couple weeks ish.
Every Wednesday.
Every other Wednesday. Thank you, Misha. We have folks joining from Europe to speak. Rosalind McAlpine and Max Wolf, I think, coming up. They're both fantastic, talking about different aspects of psychedelics and psychedelic therapy and psychedelic science. And I am delighted to have Josh Siegel here. I've known Josh for a few years. He did his MD, Ph.D. At WashU, I think, and then did a residency and maybe a mini fellowship in psychiatry. I think he spent a lot of time in St. Louis. That's what I recall. And then just a few months ago, I think you moved to NYU, or you're an assistant professor in psychiatry, a co-investigator at the Center for Psychedelic Medicine there, which we were just talking about is not as cool as Stanford's, but still, I think you're pretty cool, probably. No, the center is cooler, but the interest group is not as well developed. So anyway, thanks for your on. And he's done some amazing work in neuroimaging and biomarkers, not just with psilocybin, which is I think what you're going to talk about today, but ketamine and other rapid-acting antidepressants. There was a really cool cover story in the New York Times, I think about a year ago, when Josh's paper came out on psilocybin desynchronizing the human brain, and now we have I think a cooler image to supersede that, whatever that circle thing with all of the spaghetti in between that everyone showed before, and now we have the oscillating brain. Anyway, I'll shut up now, but Josh, thank you for joining us, and the floor is yours.
I hope so. I didn't like that spaghetti, that thing. It bothered me. So I'll take it. Mostly credit to WashU Media Relations for that, but I'll take it anyways. But anyways, that was a great introduction. Thank you. I appreciate it. And let me get my slides up.
AJ, can you mute us again?
All right. How does that look? What do you see? You see a slide, a cover slide?
It's great.
Fabulous. All right. So I will just say by way of preamble that I love to be interrupted. Please ask questions, in between. That would be great. And I've sort of made it somewhat broad, partly in hopes that you will do that people will latch on to what they think is interesting or controversial or disagree with. And I'm happy to go deeper into those subjects. So that's all I'll say to start. So a trip across levels of neuroscience, in addition to being a clever pun, the point of the title is to as I was just talking about with Boris, I consider myself a translational neuroscientist, which I think, the whole endeavor is to understand, how do we link all these things together? And that's, I suppose, what I try to do. So, from the molecular of psychedelic science all the way up to the systems and to behavior and to, clinical implications. So, disclosures, not exciting. This lecture discusses non-FDA approved treatments. That's true. And then start out by recognizing I started this work at WashU during residency, and I'm very grateful to everybody there for supporting psychedelics research and for taking a chance on the project that we did there. This is the bottom left is a picture of the first dose of psilocybin ever given in Missouri, or like that. It's like superlative or claim to fame. It's kind of fun. Um, and this is some of the folks that I've worked with to do this stuff. So, all right, my sort of big conceptual question to start off with is how can a molecule that acts for a few hours produce a change in how someone feels and behaves that lasts for months? And uh, not only that, but how can we understand that change and integrate that change across what's happening at the cellular level, where there is this fascinating observation of change that seems to last for weeks or maybe months, up to the systems, to the behavioral level, and so that's going to be the focus of the talk. And then the bonus is the other sort of thing that I spend my time thinking about, or the lion's share of my time thinking about, which is the potential of what is called precision functional mapping in neuroscience and CNS drug development. So I have some slides on that at the end. But if people are so excited to argue about the psychedelic stuff, then we'll see if we get to that. All right. So I'm gonna also go through the background pretty quickly because I'm gonna assume that folks have familiarity with what a psychedelic is and with why there is interest in psychedelics. So I'm gonna skip over this. Just suffice to say that there's been promising clinical results with psilocybin. This is the largest published trial, I believe, still to date. And I do like this quote from a trial participant because I think it's emblematic of some of the deeper ideas that we're going to get into. It was a change of state to be stuck in that place of rumination and to be able to move out. It reconfigures you somehow. And so what's going on here? And then I also have to acknowledge my new colleagues at NYU. So this was the largest addiction study published to date, and this was with alcohol use disorder and with psilocybin. And I'll use this just to make the point that, we're not only talking about promising results in depression, we're talking about promising results in addiction and in a range of other psychiatric illnesses that are being explored further. So this leads us to the question, how do psychedelics produce lasting change? And I'm going to present a few different models, or I will say ideas about how to answer this question. And to come back to, what I started with, the levels of neuroscience, I think this kind of nicely frames. This is a figure actually from a paper that I studied this interdisciplinary major in undergrad, which was like philosophy of neuroscience, and we one of the focuses I remember was this paper by Churchland and Sidnowski, where they talk about understanding the nervous system at these different levels, and that ultimately the levels have to integrate. So that's why I like this model. But the way I'm using it here is to frame some of the stories that have been building in the field of psychedelic science, to understand what is going on and why, what is the therapeutic mechanism of these drugs. So, answer one is that it produces a transformational mystical experience, and I'm just showing very simply data from uh, Griffith's 2006 paper showing that, when you have people take a high dose of psilocybin in this context, you get results kind of like this, which is that the majority report that it's a mystical experience, the majority report that it's one of the most meaningful experiences of their lifetime, and the majority report um, lasting increases in life satisfaction and social relationships. So that's been sort of the classic story about why these drugs are therapeutic, is this experience that they produce leads to lasting change in mindset. That was sort of the main focus of the Michael Pollan book, How to Change Your Mind. Oh, I'm going to skip over that. Hold on. My dog keeps scratching the door to come in. So I got to let my dog in. This is... Hold on. All right. So that's story one. Story two is, which many of you are very intimately familiar with, is at the synapse and molecular level, is a story about neurotrophic factors and about synaptogenesis and plasticity. And I'm going to breeze through that too, because I know that you all are probably pretty familiar with it at this point. We have the neurotrophic hypothesis, which states that stress induces um, atrophy of pyramidal neurons in these, in multiple parts of the brain, but probably the most studied and apparently clinically relevant are the hippocampus and medial prefrontal cortex. And antidepressants recover the deficit of, synaptogenesis and of spinogenesis and of dendritic spines. And this seems to be sort of maybe not universal, but true of many antidepressants, ketamine and psychedelics being no exception. And ketamine and psychedelics rapidly produce this neurotrophic effect. And not only that, but with ketamine and psychedelics, with psilocybin, we'll say in particular, you have this neurotrophic effect that lasts for weeks. So this seems like a great candidate for why these drugs are producing clinical response that lasts for weeks, for a week or two in the case of ketamine, and for perhaps, longer than a month in the case of psilocybin. And that's all very exciting. And I certainly it's a unfinished story, but still a compelling story that I think has enormous importance. But what it doesn't tell us is what does that mean? And how can we understand that? Is it happening in a human brain? What are the sort of uh, implications of that, of synaptogenesis and plasticity at the level of networks and maps and systems? And that was sort of our main focus when we started the psychedelics, the program in psychedelics research at WashU. And here's a list of some of the questions that we thought we could answer that we felt had not been adequately answered. So linking synaptic plasticity to circuit level plasticity and network dynamics, and understanding the neurobiology of the mystical experience, which you cannot study very easily in rodents, and what brain circuits underlie subjective and therapeutic effects of psychedelics. Are they the same circuits? Are they different circuits? This is a very important question. And then a range of other things, like how does context affect the neurobiology of the interaction between pharmacology and neurobiology. And so these are questions that doing human imaging and human biomarker research is well suited to answer. So, oh yes, so I've started that work there, and then it was actually a little over a year ago, transitioned to an assistant professor job at NYU, where I'm doing similar sorts of work. So That's what the most of the rest of the talk is digging into these questions. Okay, so only 10 minutes wasted on intro. Now we can get to the fun stuff. And again, I'm going to remind you, please interrupt if you have questions or want to argue or something's confusing. Be my guest. Okay, so a little background of the methods. I lied. We're not going to go to results just yet, but I will go through this pretty quickly also, because probably most of you are relatively familiar with resting state functional connectivity. So a lot of the results I'm going to show you use this approach where you have somebody lay in the scanner and we're imaging the bold signal, which is fluctuations in blood flow at rest. And from that, you can create this connectivity matrix between how correlated one region is with another. And this is many regions with many other regions. And from this, we can define brain networks, which we have seen over the last 20 years or so, highly, related to functional systems in the brain and sort of recapitulate important functional groupings. And the problem with this approach is that conventional fMRI is too noisy and variable for circuit-specific drug-level inferences. And specifically, what I mean by that is that there is a high degree of functional variability. So I like to use the analogy of faces. Individual faces are different, and if you wanted to study, let's say, how facial expressions changed, you would not do that by averaging faces together and then looking at, the change of the average face over the course of some intervention. That would be a silly way to do it. Instead, you'd probably want to look at individual faces and understand how the features of individual faces change over the course of the intervention, and then average together your results at the end. And so this has been a large part of the problem with fMRI studies and part of the reason why they have been notoriously uh, underpowered or requiring thousands of participants. And here's a nice example of that. Very simply, what I'm showing here is a contrast between it's an fMRI, sort of classic fMRI design. You have people look at scenes and look at faces, and then you say which part of the brain activates to scenes more than faces. And here's one subject in the study, and this yellow area is the part of the brain that activates the most to faces, to scenes rather, more than faces. And then I'm showing outlined in white defined by completely independent resting data in this same individual, this resting state network called the context network, which we know is related to viewing scenes. Okay, so that's one person, all well and good. Here's another person, all well and good. This is the part of the second human's brain that activates to scenes more than faces. And this is from independent resting data, this person's context network. So great, but when you look at network, the network from human 2 on the task activation from human 1, you see that they are very poorly aligned. And this is constantly the case in fMRI studies. And the consequence is that when you do group averaging, which is how we do almost all fMRI studies, both task and resting, you get this much more blunted activation of scenes greater than faces. And that activation is not only much weaker, but it's also not actually it's losing a lot of the, spatial topography, and it's not actually representing any individual, and it's also a lot weaker. So this is a problematic approach. And here's empirical data with, sort of demonstrating this in the context of drug studies and showing that how much does connectivity change between scans within a session? How much does connectivity change between sessions? Or how much of overall variability in connectivity is attributed to change between sessions? How much overall variability is attributed to drug effect? In this case, this is stimulant. And then how much variability of connectivity is attributed to individual differences, and it's much larger than the drug effect. And so if you can control for individual differences, this becomes a much larger percent of overall change, and you suddenly have a much more powerful tool. So this is basically the idea behind a lot of the work that has gone on at WashU that I have had the fortune of being a part of for some of it, developing what's become known as precision functional mapping, which is I defining these networks in individuals rather than using the group average. And this has proven to be a very powerful tool and as I said, made fMRI potentially a much stronger biomarker. And so I sort of conceptualized taking the next step with that with PFM in drug trials, which is using within individual design really to look at the effect of an intervention. So not only using individualized precision functional mapping of networks, so rather than saying, how does coordinate XYZ change in its connectivity to some other place, saying how does the default mode network in this individual change with respect to some other place, for example, but also, using a longitudinal and an individual analysis. So this is a really important concept. So that's why I've taken a minute to really hone into that. And then I'm going to use it to introduce the psilocybin study that I worked on at WashU. And here's just an inspiration for that study. So the Nico Dosenbach's lab had previously done this CAST study and used a design with very intense within individual imaging. These are all fMRIs. So basically, they had somebody get in the scanner every day for 42 days. And in the middle of that, they put on a cast for 2 weeks. And what they saw was that when you had this much data on an individual and you did, precision identification of individual areas, you got this huge effect that's bigger than any fMRI study effect that I'm pretty sure that I've ever seen. So that was sort of the inspiration for the psilocybin study that we designed. And in the case of the psilocybin study, it was a crossover between psilocybin and methylphenidate, which is Ritalin, and it was chosen to match the arousal effects of a high dose of psilocybin. And it included multiple baseline scans and then scanning on drug and then multiple scans between and then scanning on drug two, multiple scans after, and follow up a few months later in a repeat drug dose. And this is just showing, we use this approach to individually define brain areas rather than sort of group averaging. I think I'm gonna just lose my voice if I don't have some water. So I'm gonna take a 10 seconds to go get a cup of water and I'll be right back. Okay? Any questions before I go? Boris says thumbs up. I'll be right back. I'll be right back. Right. Great. So on we go. Unless there's questions. All right, so what do we find with this psilocybin study? What we found, I'm going to start out by saying that what we found is that psilocybin massively alters functional connectivity in multiple parts of the brain, including multiple parts of the cortex and multiple parts of you see the hippocampus below that and cerebellum and medial thalamus among the areas most affected. And then you can see the effect of methylphenidate for comparison. And so this was a very large change, and it so happened that the network that was most affected by psilocybin is the default mode network, and we are not the first group ever to report this, but this is certainly what we found in our data, and just to give you a brief summary of what the default mode network does, it's implicated in self, introspection, theory of mind, and it interacts with the hippocampus to create our sense of self in time and space. And it's often been associated with mood disorders and been, been reported to have abnormalities in rumination and various other psychiatric illnesses. And what we found was that people's report of on the mystical experience questionnaire, which we use to assess people's experience afterwards, was very strongly correlated with the magnitude of change to connectivity in the default mode network. And in fact, if you look,
Josh,
yeah,
I have a question. So that the mystical experience, that relationship, people have measured MEQ against a large number of outcomes, and this is probably the strongest correlation I've ever seen. Am I just misremembering, or is that actually accurate?
no, I think this is stronger than most, yeah, MRI correlates that have been found previously. In fact, this is probably why it got accepted to Nature because I didn't even think to look at the correlation. This is a side story, but I'm just going to tell it anyways. I didn't even think to look at the correlation between people's rating on the MEQ and brain change, which maybe is stupid of me, until had submitted it already and a reviewer asked, and then like that became the most exciting result in the paper. So then it couldn't reject it at that point. But yes, it was a very strong correlation.
And can you just for including myself, like when you say it massively alters functional connectivity, can you contextualize that at all, like compared to what?
Yeah. That's an annoying word. I know. I'm sorry, but the reason for making a statement like that is I guess I'll put it this way. So, methylphenidate or Ritalin, we think of it as one of the more powerful drugs in psychiatry. It has noticeable changes in cognition and behavior. We gave people a pretty good dose, but relative to how much methylphenidate changed connectivity, the effect of psilocybin was 3 or 4 times larger. So that's one, maybe way of contextualizing it. And then there's some other pieces of sort of evidence about the magnitude of the effect. It's a descriptor of magnitude of effect, but the point being that many ways that we measured it, it was a very large change, far beyond sort of day-to-day variance or what could be seen with any other manipulation. Okay, so you can uh, you can complain about the word if you want, but that's the reason for choosing. And then any other questions? All right, I'm gonna keep going. What I'm showing here in this little inset is that specifically, uh, actually this is desynchronization, which I haven't shown yet. So that's a little bit different than functional connectivity change. But desynchronization of signal within the default mode network specifically is most strongly correlated with disruption of with one subscale of the 4 subscales of the MEQ, which are all totaled here, and that subscale is change in perception of time and space. So I think that's also interesting and comes back to what's in here regarding how we think about the function of the default mode network, and I think there's a lot of intuitive logic that disrupting function in the default mode network alters the sense of time and space. So okay, but that's just an observation, and I'm mostly presenting it to set up the question of why is there such a large change in functional connectivity, or why is there a large change in functional connectivity in general, and what's going on, and how can we interpret it? And so the first thing I want to do to try to get at that is go back to the sort of square one and just look at the signal in the resting fMRI brain And what I'm showing on the left here is placebo, and this is just one subject, one scan, and on the right is psilocybin. And you know, I'm sure most of you aren't used to looking at videos of sped up videos of brains fluctuating, um, but I'll just sort of pause it and can maybe make a point about it, which is under normal conditions, so placebo over here, the characteristic of resting fMRI is that you have organized activity, and in general, you have sort of collections of brain areas. So the areas, so the red to yellow is greater bold signal, and the blue to green is lower bold signal, below the sort of mean. And the classic observation with resting fMRI is collections of areas that co-activate and deactivate together. So this is like, I don't know, probably the frontoparietal network in this individual. And what you can see with psilocybin, if you spend enough time staring at these videos, is that there's just much less spatial organization to the signal. And so, so when I saw the functional connectivity change, I went back and looked at just sort of looked at these videos of what is going on with the bold signal. And then sort of secondarily, I said, well, how can I quantify what I'm seeing? This looks much more less spatially organized. And even when you play the video, it looks less temporally organized. And so we came up with this measure, which we didn't invent it. It has been published with fMRI and EEG studies of desynchronization called normalized global spatial complexity. Now, I will say I think it probably has a lot of overlap with ideas like entropy and the entropic brain hypothesis that Robin Carhart-Harris and others have put forward. Um, the normalized global spatial complexity, the name kind of, I think, does a fairly adequate job of describing what it is. So it's just how spatially complex the signal is over time. And this is the 6 participants in our study, and for each participant, their psilocybin NGSC scores are the black circles, and the non-drug NGSC scores are the empty gray circles. And so you can see very consistently you have this greater desynchronization on psilocybin of brain activity than non-drug scans. And this is across the whole brain, but there's actually a topography, and I'm showing that here. Not surprisingly, it nicely matches the default mode network, and it nicely matches the topography of areas where I just showed you previously that functional connectivity is disrupted on psilocybin. So that's story one. So now at least I think we've peeled back the onion a little bit and showed, what does the functional connectivity change actually mean? What it actually, or what's it actually measuring? Well, one answer to that question is is that the local signal within a brain area is much less organized and structured than on placebo or on no drug. So the other thing that I really like about that story is that it's actually been observed very consistently across psychedelic studies, which, to come back to this idea of sort of levels of neuroscience, it's very similar observations have been made all the way down to the unit, to single unit recording studies. And so what I'm showing here on the left is that studies, and this is by Chelada 2008, they measured local field potential and single unit recording. And in baseline conditions, the majority of single unit firing of pyramidal, this is pyramidal neurons in the prefrontal cortex. The majority of firing is during the active, aligns with the active phase of the local field. But under a psychedelic, actually, there is much less preference for the active phase. So depolarization happens much less often when the local field is in the active phase. So this is quantifying that and just showing percent of spikes during the active phase is much lower on the psychedelic DOI. And then at the mesoscale, there's been very similar observations with entropy and Lempel-Ziv complexity, which similarly assay sort of the organization and structure and desynchronization of the signal. Yeah, so this is from Timmerman 2023, using this Lempel-Ziv complexity and really, I think, a very parallel observation with EEG, which is nice because that suggests this is not just a blood flow phenomenon, right? You're seeing desynchronization, and they also show that they look at the richness of experience, which is one of the qu questions that they had people rate in that study. This is actually with DMT, but it correlated strongly with increases in their measure of complexity, which is fairly similar to desynchronization. Okay, so that's a nice story about what desynchronization, what's leading to the functional connectivity change. There is another story that I have worked on with collaborators at Stanford, the fantastic lab um, of Leanne Williams and her postdoc Adam Pines, and they have specifically looked at the flow of information on psychedelics and how the how the flow of activity across the cortex might be changing, and whether that helps us understand both the desynchronization and the change in functional connectivity that has been observed. And part of the reason that this is really cool work is because, oh, where my animation got all screwed up. Oh, there we go. I had it in the wrong order. Okay. Part of the reason that this is really great work is because they have combined multiple data sets, including MDMA, psilocybin, LSD in humans and LSD in mice. And so they've been able to really powerfully, expand and other drugs in mice as well, and look at these, questions of activity movement or optical flow. Um, across different drugs. And what Adam has found, and this work is still under review, is that uh, the flow of activity, which has a general preference to go from primary cortex to higher association cortex, um, and has a general preference to go into the DMN, the default mode network, uh, which is why there's positive values here on placebo. But that flow of information, bottom up flow, we'll say, decreases with MDMA. The bottom up flow decreases with psilocybin, and the bottom up flow into the DMN decreases with LSD. Uh, the same result across humans and mice, and really a nice observation. I'm showing here that not only the same result of decrease in the magnitude of bottom up flow into the default mode network on a psychedelic in mice, but actually no change or the opposite change with diazepam and dexmedetomidine. So at least to some degree, drug class specific observation. So that is a really interesting story that's still unfolding. Understanding, how the flow of information across the cortex might be changing and how that might be producing some of the psychedelic experience. Um, and then uh, one area I know Boris has been interested in, which is the third story about how can we understand the changes in functional connectivity that we observed with psychedelics. And so I want to say, in my defense, that from the time that we were designing these, studies and these human studies, I was really immersing myself in the psychedelic literature, and part of that was coming across the observation that the 5-HT2A receptor is in astrocytes. There's, there's known effects on the vasculature and vasoactive effects of psychedelics. And so one of the really important questions to me was, to what extent are the changes that have been observed in fMRI studies, the many that came before me included, attributable to change in neurovascular coupling or just sort of neural effects of the drug? Because of course, with fMRI, it's quite difficult to differentiate the two. So I take part credit for the idea, but no credit for executing this study, which the entire credit for that goes to Jonah Padawar-Curry and Adam Bauer, who I'm showing down there at the bottom left. And they have this great setup where they can simultaneously measure basically blood flow changes with FCIOS and calcium fluorescence from neural activity from pyramidal neuron depolarization. And so that's what's being shown here. And so we used this setup to basically compare changes in neural activity assayed more directly to changes in blood flow. And I'm not going to go super deep into it. This study just came out a few days ago, actually, in Nature Neuroscience. So I would encourage you to check it out. But I will just give you the sort of one key boiled down takeaway point, which is that when you look at, so what I'm showing here is the impulse response function. In other words, the mathematical relationship between neural activity and change in blood flow. And so by measuring both simultaneously, we can actually model that relationship sort of without any priors. And so this is what the relationship looks like before and after saline, and then before and after the psychedelic DOI. And before is in red, and then after is in blue. And there's two sort of things to take away from this. One, it looks pretty similar. And the other, it's significantly changed, which the change is that the full width half max, or in other words, the width of the hemodynamic response to neural activity is smaller after DOI. So this up and down response is a little bit narrower after DOI, which I have to say, 2 points about that. One is not what I expected, and two is remarkably consistent across task data, rest data, and even pretty consistent with human data. And so here's data from that I collected from humans, and this is just showing two areas in a task fMRI study. And you have left visual cortex and left auditory cortex. And when you model the width of the hemodynamic response, you can see that in both areas it's significantly decreased. So we used a 3-parameter model, and the D parameter is the dispersion. That's essentially very similar to the full width half max. And in these areas and in many other areas that we looked, the dispersion parameter was decreased on psilocybin. So very interesting. And, again, not enormous change, but a significant change. Okay, so that is all I'm going to say about acute changes during a psychedelic. There is a lot more that could be said, but I've left it out and invite people to ask questions if they're curious about, other sort of investigations into how brain signals are changing acutely. But the last thing I will say here on this topic is uh, in this data set, really is what I should say, is that we also looked at lasting changes in the weeks after psilocybin. And the observation that we made, in fact, specifically, we had looked across the literature at what had been observed in the weeks afterwards and where the biggest neurotrophic changes are. And I think I mentioned this earlier, that anterior hippocampus, medial prefrontal cortex as well. So those are the areas that where there appear to be neurotrophic effects that are correlated with clinical response. And so that's where we looked for connectivity changes that last in the weeks after, with the idea that, if there is a clinical response that's lasting for weeks, there should be a neurobiological correlate of that clinical change. And what I'm showing here, this is actually from a review, which is describing this circuit between the anterior hippocampus and the default mode network in cortex, and specifically describing its role in self-referential memory. So interesting implication or function, self-referential memory. And what we observed is that acutely on psilocybin, there's a large change in functional connectivity in the anterior hippocampus. And afterwards, smaller, but still significant for weeks afterwards in this anterior hippocampus. So I know this is weird to look at, but this is the amygdala, this is anterior hippocampus, this is posterior hippocampus. This is just a axial slice on an MRI. Overlaid with this functional connectivity change measure. So what is this change? What it is that if you put a seed here, you see that the connectivity with the cortex, and in particular with the, so normally this area is connected to the default mode network, which is why it's shown in red here. But after psilocybin, the connectivity with the default mode network decreased. And in fact, pretty consistently, so this is the 6 participants in the study. For each one, pre-psilocybin scans are on the left and post are on the psilocybin are on the right. And so you can see pretty consistently there was a small but reliable decrease in connectivity between the anterior hippocampus and the cortex. And here's one example from one individual, and you can see the areas where there's decreased connectivity in blue, and then I've overlaid on top of that the borders of the default mode network. And so there does seem to be not only, but definitely a decrease in connectivity between the anterior hippocampus and the default mode network. Now, this is an image from a preprint that just came out maybe a couple months ago from Alex Kwan's group, and the goal of this study was to try to bridge the connection between what's happening in the brain acutely with a psychedelic and what are the lasting connectivity and plasticity effects, and how can we link those two things, which I think is, one of the most interesting and important questions that we're asking in the field right now. And the reason I bring up a small part of a figure from that paper is because one of the things they observed is that connectivity between their cortical region, which I believe is like precuneus, which is mouse default mode network, and many limbic areas, including parts of the hippocampus, was decreased, and that seemed to relate to a decrease in activity in these areas on psilocybin. So, I think still very different and complicated, but to some degree seems to recapitulate this observation that there is a decrease in connectivity between hippocampus and cortical areas that may be sort of a circuit level correlate of some of the neurotrophic effects, lasting effects of psychedelics. So, one really interesting but speculative link here is that the acute desynchronization, or possibly, you can describe it however you want, perhaps the flow, activity flow, is what's triggering a burst of plasticity and then leading to this lasting reset of circuits that might be related to psychiatric illness or depression. Okay, I'm going to pause here. I have some more slides, but they're about the potential of precision functional mapping in fMRI as a tool for better understanding drugs. And so they're not about psychedelics per se. So I'm going to pause here and open it up to folks.
Josh, I got a question. Acute. So what other things will acutely desynchronize brain? And I'm thinking about, what do we see in operating rooms, when we're monitoring return to awareness, it just sort of looks like, all that sensory information that was not coming through now is suddenly coming through, and these well-organized rhythms of unconsciousness where you're basically keeping the soup warm, but nothing is actually going through. Now it's receiving all this information. So are you saying something fundamentally different than more sensory input good? Like,
Well, it's brilliant that you picked up on that parallel so quickly. I think that you're getting right at the way that I think about it, which is that certainly one thing that desynchronizes the brain is stimulus or engagement in a task. So, I mean, a lot of the way that I've thought about this connection is by literature showing that, task-induced desynchronization triggers, experience-dependent plasticity. And so certainly I think of this as parallel, and there's probably a lot of sort of other implications of what you're saying that I haven't thought through yet. And so I think it's a compelling idea. But yeah, I kind of do think that this sort of desynchronization is it's not necessarily a psychedelic specific thing. It's some sort of, you know, correlate of I mean, I think you could draw on these entropy ideas that this is sort of the signal of firing in an unfamiliar way is fundamentally like the Hebbian plasticity mechanism. And when the brain starts, goes from being highly synchronized and organized to a more disorganized or differently organized state, that's what triggers some of these, neurotrophic mechanisms.
So another counter example would be I don't know if it's a good one, is salvia, right? So there's a Hopkins Binoche dose had a paper from Hopkins where I may be getting the details wrong. He gave a whole bunch of drugs, one of which was salvia, which we don't typically think of as therapeutic. Maybe that's a mistake, but the conclusion that he drew was that all of these drugs will desynchronize or maybe reduce coupling from the DMN.
Yeah. Yeah.
And again, it just seems like that's all we're really measuring is engagement of the CNS with more like just stuff, input of any kind, that it doesn't really have valence or even this idea of plasticity. We're still missing that causal link of if you were to desynchronize the cortex and do nothing else, right? Independent of experience somehow, with a bunch of phosphines or white noise. I'm pretty sure that would desynchronize something. Then would you? I mean that would be one way that you could kind of get at that. And I think your prediction and mine would be that wouldn't be either therapeutic or lead to any sort of long-term change.
Yes. I think, obviously the question is where is this happening? And in the case of psilocybin, it appears to be in many places of the cortex to a high degree. So, where and to what magnitude? So many places of the cortex to a high degree. And my hypothesis would be that like a very specific, stimulation or learning protocol would induce desynchronization. I mean, just like we know that, giving visual input desynchronizes the visual cortex, and giving visual input is the impetus for visual learning. So, obviously it depends where, obviously, but it also depends, what is the context, what is the stimulation, what's happening afterwards. And so, so I don't want to oversell this idea, but I think I view it as sort of an impetus for exactly these types of studies that the Quan Lab, is trying to embark on to better understand what is it about the acute change that is inducing lasting neurotrophic effects.
And you didn't see anything like that. I think you showed it, and I missed it, but with methylphenidate, which is also like pretty engaging, but the acute effects were not. I guess can you just remind me a little bit more about like how are they different?
Yeah, so I mean there was certainly no desynchronization at all, which I don't think I show necessarily very clearly here. This is just showing, the magnitude of functional connectivity change. And this pattern with methylphenidate in, primary motor and sensory cortex and lateral, is like area MT. Uh this is like a classic arousal pattern. So there is connectivity changes related to arousal. There is not desynchronization at all. Um, there was no lasting change observed. No lasting. I mean, we didn't look everywhere in the brain, but in the places that we looked, the hippocampus being one of them, there was no lasting effect of a single dose of methylphenidate.
And the prediction would be if you could if you could get your numbers up, desynchronize harder or whatever, if you gave enough methylphenidate, at some point you would get more red and not just green, and then maybe achieve the same.
Well, no,
lasting.
no, no. I mean, I want to be careful about language. Functional connectivity change is you take area A and area B and you look at how correlated their signals are over time. Desynchronization is something different. What I'm referring to with desynchronization is within a collection of neurons that are normally fluctuating in synchrony, they are going out of synchrony, okay, within a brain area. So that's not happening. So these things maybe are easy to conflate, but there is a change, there is a decrease in connectivity in these areas with methylphenidate, but there's no desynchronization.
Other questions in the audience? Questions in the room?
Josh, I have a question about plasticity. I don't know if you can see me over here, but when you go back to the slide that you ended on, or that you talked about how desynchronization would cause plasticity. Like more disorganization would create more plasticity, which would then change circuits. Is this like mainly LTD, like a reduction in new connections? Because maybe like my way of thinking about this is like neurons fire together, wire together. The more regular the firing, the more plasticity is happening. And it seems like you're saying almost the exact opposite, or maybe it's like the reverse, like LTD is happening.
Yeah, good question. No, very good pickup and good question. And, all I can say is, this is a speculation based off of what I'm observing in bold data. So, for one thing, I can't confidently, I mean, there is areas that, let's say, become more correlated with a psychedelic that were not previously correlated. So there are areas that become less correlated and areas that become more correlated. But I can't, I don't want to like overstate this idea. I also can't definitively say if, if this connectivity changes, it would be reasonable assertion of that LTD is, for example, related to this lasting loss of connectivity between the hippocampus and the default mode network. But we also know that there's a lot of LTP that's happening and synaptogenesis after psychedelics. So all I can say is there are better tools that I think would be have to leverage to answer that clearly.
Thank you, Josh. Any other comments? I have one more. This is a little bit unrelated, but kind of related to the whole, mimicking the subjective effect. I don't know if you work with those folks now at NYU. They use 50 to 100 milligrams of Benadryl as a control. You're using something a little bit different. This is a kind of a pressing question of how do you, come up with a good control. Have you heard anything about diphenhydramine as either mysticality or otherwise, or has anyone ever put people who are delirious from Benadryl in a scanner?
It's not a good control. It's 95% of people correctly guess which drug they're getting. And no, I'm not sure what the effects are on
Right.
brain activity. And we'll
Maybe
go
they
to
should-
the last slide. Last slide. This is also ongoing projects and collaborations that I have going on, which happy to chat about those as well. So the thing I didn't get to was the problem with group averaging. I did a little bit in the background, and further exploration of how it can improve drug targeting, not drug targeting, drug biomarker studies, and how fMRI, if done optimally, can be a much more sensitive tool for neuropsychiatric biomarkers. And then here's some other projects that I'm involved with.
Josh, I want to thank you again. I know it's late in New York.
Yeah, this time worked out well. It's perfect.
Excellent.
The kids are already in bed.
Awesome. We'd love to have you out in person, the next iteration of this, but this is really, it's amazing work. I'm more convinced than I was at the beginning of the hour that there's something going on there. And it makes me want to reread some of those papers, but thanks again, for joining us, and the rest of you all, I hope we'll see you in two weeks, I think, whatever the next one is.
Thanks a lot for the invitation. And I'm going to have to get on the mailing list because definitely want to check some
Yeah,
more.
come by. Have you as heckler. Or not heckler. Anyway, all right. Thanks, Josh.
You're welcome.
Thank you. Thank you everyone for joining.